Really? Is it on? Okay. Listen, I'm going to do a very short introduction. As you see, I didn't have time to put on my professor costume today, as I sometimes do. And that's because Markus and I had a great conversation for the last hour or so, talking about a wide-ranging array of topics. The discussion was not only so good that I didn't get to change, but it was also so good that I didn't do the normal interview that you do right before you interview somebody, where you ask them, you know, blah, blah, blah. So let me tell you the parts that I know. I know that Markus was a postdoc with George Koob many moons ago over at Scripps. And I know that somehow then you made your way over to NIAAA, where you were intramural for a number of years. And then about 3.5 years ago, you moved to an unpronounceable part of Sweden, where you currently run a large center. And we may share a kind of a common interest in disrupting little branches of fields by putting out inconvenient truths about them. And we talked about that over the last hour. So he's going to pass on some inconvenient truths to you. And if you want, we could do like an interview and I could find out the rest of your academic history. It's, yeah, okay.
No one could care less.
It's, okay, perfect. He knows it, and the rest of us would likely forget it. So
yes.
I'm going to turn it over to you so as not to steal any more of your time. Sorry for the informality.
Thank you so much. So I enjoy these little informal gatherings. Please interrupt me whenever. It's always fun to come back to these parts for many reasons, not the least. It's actually bright outside more than 5 minutes. This was an interesting trip, though. You know, when they tell you that at some point in your career, maybe you'll be giving keynote lectures, I didn't understand that that meant that you'll have to use some crappy software from Apple called Keynote to recreate talks that disappeared on the computer that they stole for you at Newark Airport, which is what happened yesterday. So if that, just to explain that if there are some fonts and stuff that look weird, that's because of the various conversions that have had to take place since to recreate the talk. We have a talk. So, all right. I'll be serious for the most part from now on. This is a title that some people get upset over. So, you know, let me just point out that at least I got things wrong, and I'll explain to you how and what we're trying to do about it. I can also tell you, some of you probably know this, others less so, but as painful and disruptive as it is to move, as good it is for reinventing your thinking. Because basically I moved back from the NIH, where I had spent over a decade leading the intramural translational research program, back to Sweden 3.5, almost 4 years ago now. And I was beginning to realize that even though by academic standards we had been very successful in our program, publishing well, and people seemed to be happy with us, I was frustrated out of my brain because the things I really cared about accomplishing, which is bringing effective treatments to patients based on the science we do, just wasn't happening. And so for the past 5 years, I guess, because this started brewing in my head even before leaving the NIH and was one of the contributing reasons for me to decide to try something new, was I wanted to rethink our ways. So whenever I criticize the way things are done, I'm only criticizing the ways my lab has been doing it, okay, just so you know. Okay, before I move on, when I was at the Karolinska, which was 1 decade of my life after that postdoc with George, and then at the NIH, another decade plus, I never had to show maps of where I am. So that's what comes with moving to a place called Linköping. Yes, it can be pronounced, which is here. And unfortunately, these laser pointers get absorbed into these types of screens, so they're completely pointless. So you'll just have to, is there an old-fashioned, like something like...
Can we get that, like a radio antenna?
Yeah.
You would break the antenna off a broken radio and use that. But...
Well, if not, then don't worry. Oh, there it is. Oh, look, I love these. So this is Linköping, 2 hours south of Stockholm, the smallest medical school in Sweden, and we now have this place, which is about 60 people, 5-6 different research groups, studying all kinds of weird things, ranging from rats and mice that I'll be talking about today, human imaging and experimental medicine, but also weird things like the neurophysiology of social touch and things like that, which I will not be talking about. But we truly try to do things that move things around and see if they will come back together in new ways. And also, I had those great ambitions for building European networks when I moved back, and of course that's kind of interesting because some of our best friends are basically disappearing off the map right now, so that's what you see there. Okay, this is just, I'm not an epidemiologist. My impression of the disease burden from alcohol is shaped by very different experiences than reading the numbers. It's from treating patients over a career as a physician for close to 30 years now, but it truly is one of the greatest contributors to an amount of misery that's hard to imagine. This is the backdrop, but the other part of this is it's also one of the most unmet medical needs in all of clinical medicine. Even the medications we have that have the indication are prescribed to less than 3-4% of patients with established diagnosis of alcohol use disorder, at best in specialized services, okay? So that's sort of the background of the amount of unmet medical needs. So we clearly need to do something about it, and the idea was that we were going to, because of course the explosion of modern neuroscience was going to address these things. I actually started my postdoc with George here. At that time, I was not actually an addiction guy. I was a stress and anxiety guy, an amygdala guy, I guess. I took a look, because the other half of George's lab was doing addiction, and I took a look at alcohol addiction research, and I decided against having anything to do with it, because honestly, in those days, it wasn't all that interesting, because a lot of it was studying organ damage from alcohol, which is clinically obviously tremendously important, but it's not the solution to the problem of alcohol addiction. Since then, of course, it's been just fantastic. If you put on your neuroscientist hat, the quantitative explosion has been exponential, which this is the number of papers that can claim to be papers trying to address mechanisms of alcohol addiction using neuroscience strategies. But it's not just the quantity, it's also this level of sophistication with which we go about this. So in those early days when I came back from my postdoc and started up my own lab, we could poke holes in rat amygdala with some ibotenic acid, then put the rats in some cages and measure how much they drank for a few weeks, and we'd have a very good paper. Actually, one of my better cited papers. All these years later, the field has developed these tools that allow us to not only tag ensembles of neurons whose activity is associated with very complex behaviors, but also control the activity of those ensembles using optogenetics or chemogenetics in ways that actually allow us to finally nail down causality. It's fantastic, right? So if it were from the scientific perspective, it's been a great ride. Except I then turn around, put on my white coat, and go out and keep treating patients with drugs that were serendipitously discovered 50 years ago, before any of this was even conceived. Okay? And not that I'm paranoid or anything, but this is the year I came to the NIH to build up a translational program with the objective of developing neuroscience-based treatments for alcohol addiction. And at least until that date, there were 3 that had gotten approval from the FDA. And I hope it's not me, but since the day I embarked on this expedition, the number is 0. So I personally, at least, don't feel like my endeavor has been particularly successful. And of course, no one has expressed this more vocally than Steve Hyman, the former director of NIMH, who rubs a lot of people the wrong way, but is also one of the smartest guys around, and cannot be argued with when it comes to the fact that the failures have been so many that one of the end results is that pretty much all of pharma has pulled out of developing psychiatric medications altogether. Addiction, of course, being at the bottom of the totem pole to begin with, was the first to go. And by now, J&J is still out there, and I admire them for it, and I really hope for them to be successful, and we celebrated esketamine with champagne over at our place, but that's them. Pfizer shut down the neuroscience division over a year ago, and that's just, so the powerhouses of psychiatric neuroscience are gone as a result of the failure of the neuroscience we have been doing to bring forward useful treatments. So, okay, if that's not enough reason to rethink at least my ways, I don't know what would be. So we've been trying to rethink our ways. One thing that was the first to jump out was, since Roy Wise in 85 or so published the psychostimulant theory of addiction, we've pretty much had a canonical model of what addiction is. Addiction is that animals, or modeling addiction is that animals will self-administer any drug that people get addicted to, and the way to model that is putting them in an operant box, and if they press a lever to deliver a reinforcer, that is a valid model of addiction. And Roy still maintains that that is what we should be using. So, I'll come to why I've come to conclude that that's just fundamentally flawed, but I also want to point out that I never have any original ideas of my own. I just love the people who have them, and I'd like to just highlight two people who in particular have been influential. So I don't know that Serge Ahmed invented the paradigm of alternative rewards being important. There's this hilarious video of a guy named Sprague in 1949 getting a chimpanzee to choose between one button that will deliver a box with a banana or another one that will deliver a syringe with morphine, and you can see how choice preferences shift based on whether the chimpanzee is in withdrawal from opioids or not. And then that goes through the work of Joe Brady at Hopkins in the 70s, but really Serge has been extremely influential in shaping the thinking and applying it. And then, of course, the other one is my good friend Pierre V. Piazza. And so these are two major conceptual influences. So the three things that I think we have to rethink first here. So we've known this forever. I get beaten up when I keep sticking with these original data from '94 from Jim Anthony, but that's the whole point. We've known this since at least then, when Jim Anthony was at Hopkins and published those data. So we've had the numbers. Only a minority of users develop addiction, right? And that minority, when it comes to even a so highly addictive a drug as heroin, is not much above 20%, for alcohol about 15%. So how can we hope to figure out how that works in that minority of individuals by studying processes that we study in groups of rats, where we treat the whole group as if all rats were the same? Secondly, as a clinician, you're struck with how addiction happens in a context which involves a progressive shift of choice preferences, at least originally, and at least most places. We hope for people to have a broad spectrum of rewards available to them, and then you see over time how their choices within that spectrum get shifted over from natural rewards and toward the drug rewards. Then the spectrum of choices available may narrow as a consequence, but that's sort of a secondary phenomenon. So the question is, how can we hope to understand how that happens in strategies that basically use one lever to deliver a single reinforcer, and then the other lever is inactive? And what annoys me the most is then we show the inactive lever presses, which are close to zero, as a control for non-specific effects of things. Come on. So some smart people have tried to sort of weave this into the research and understanding addictive processes. Rita Goldstein, I think, was early out there. So Scott Muller, when he was a postdoc with Rita, did a very nice fMRI paradigm where you can see that this is an adaptation of the classical Iowa gambling task that samples implicit choices and choice preferences. And you can see how cocaine-addicted people have this relatively pronounced preference for the cocaine-associated stimuli over the nice stuff that I would like to experience if I had some more time in La Jolla, right? But that never really trickled down into the rat research. And then the third thing is what you're struck by in clinical addiction is the continued use of drugs despite knowledge of adverse consequences. I can't tell you any patient stories really from my own practice, but we can just pull this from the media, right? So this is a reasonably well-functioning mother of two working full-time, and this is her with her hepatic failure a few years later, and despite this, despite obviously knowing that it's going to kill her, she continues to drink, and the last picture is the first Christmas after mom died. Okay, and so how can we hope to understand that process in models where rats press a lever, get a reinforcer, and nothing else happens? So again, there are attempts to model this in imaging environments. Actually, so I want to highlight Erika Grodin, who is now at UCLA, but was a graduate student in the intramural program with us, spent a heroic effort over 4 years to, you can model this in humans. So if anybody tells me you can't model it in rats, if you can't do it in humans, you should be able to do it in other species as well. So this is a classical imaging task, the monetary incentive delay task, adapted to looking at people's choices. They basically button press for points where they can earn either points toward a snack or points toward a dose of alcohol that they get to consume at the end of the experiment. They get primed with both types of rewards up front. And then, and it's actually my contribution to the study, we really shocked them. And the way it works is when they press for the respective stimulus, in the context of a green border, they know that it's safe, so that really shouldn't be influenced by whether you're a heavy drinker or light drinker, dependent, non-dependent, addicted, non-addicted, pick your terminology. But then yellow indicates that you have a 20% risk of being shocked with a shock intensity that you titrated yourself, okay? We're not that bad before. But, and at red, there's an 80% probability, if I remember correctly, that you get shocked. And lo and behold, not only do you see brain signature of this aversion-resistant working for the alcohol reward, you can also see that the responses under high-threat conditions really are very strongly correlated with the scores of compulsive drinking outside in the wild prior to running the experiment. Okay? So these things can be modeled, but we have not done so in our animal models. So, this is the patient of which you can buy 50 at a time and actually carry out your experimental medicine study in less than 4 years. Although the one I'll tell you most about actually took about 4 years, so it depends on what you want to do. So we tried to address these, as we think, limitations to the conventional modeling using rat models. And Eric Augier came to us from Serge Ahmed's lab. Serge had already done his groundbreaking work on the role of alternative reinforcers. Eric said already on the day of interviewing for his postdoc position that was what he wanted to do, but he also wanted to look at individual variation. That was what I wanted to do at the time, so we said, sure. Took him 5 years. One of the advantages of having these bigger contexts is you can support people in doing high-risk things that don't have to pay off within a single grant cycle, and we sort of think that's worth doing. So what Eric did was he took Serge Ahmed's discrete choice mutually exclusive, this discrete trial mutually exclusive choice model, which is this. First they sample, so both levers are out. They press here, alcohol comes out, they can consume it. They press here, a sweet solution comes out, they can consume it. But then once they know which lever is doing what, when they press one, they get the reinforcer, they get to consume it, but the other lever is retracted, so now it's no longer available. And long story short, this started within a batch of 50 rats where 4 rats were found to behave in interesting ways. So this was laughable, but of course we then did, by now I think we've done about 800 of these, and this is a very consistent thing. So you see a bimodal distribution where in 85% of outbred Wistar rats will just simply prefer to lever press for a sweet solution over alcohol, but there is this very constant population fraction of about 15% that will actually do the opposite, and they will press for alcohol. Okay, these guys show some pretty interesting other traits, so it really looks like a syndrome. One of those traits is they have elevated motivation to obtain alcohol, and it's selectively elevated, so no difference for a sweet solution, but you can see on conventional progressive ratio breakpoints they have higher motivation. But the more interesting thing is that even though we didn't look for specifically upfront the phenotype of aversion-resistant drug seeking and taking, it gets pulled out together with the choice. So you can see this shown two different ways. This is by quinine adulteration, which is the way the alcohol field typically does this. We basically add this bitter taste to a concentration that makes the 85% majority of the rat population that didn't choose alcohol just stop drinking. But these guys that are in the 15% minority, they just couldn't care less. Now this has some potential problems and confounds. The cocaine and heroin people like to do it using shock suppression of responding instead. That's technically very difficult with alcohol because alcohol is such a crappy reinforcer acutely, and there's this delay because it's oral. So a couple of labs, ours included, have figured out how to do it. If you're interested, I can tell you there are some technical tricks. But by the time you've figured it out, you can see that once you bump up the shock intensity, this is not a lot, really, 0.2 milliamps. In classical conflict tests for screening for anti-anxiety agents, for instance, you might go up to 0.6. So already at 0.2, the vast majority of the normal rats will just drop their self-administration, but the guys that chose alcohol over a natural reward just couldn't care less. And yes, all the controls that you might imagine we've looked at, so this is not about differences in nociception or anything else. It's really a reflection of them having this syndrome of traits. Okay, so one sanity check. So George Koob's lab, my own lab, Howard Becker, others for probably over 20 years now have been inducing alcohol dependence in rats, and found that if you do so passively, and this is a little bit different compared to cocaine and heroin, it's a big debate, but with alcohol, really, if you impose alcohol dependence, physical alcohol dependence long enough on a particular exposure schedule, so it's got to be intermittent and long enough and all that, you induce a bunch of behavioral traits that persist. And we've done a lot of gene expression regulation work on this, and this is not the topic of today's talk. But you can see that this is really just two data sets to illustrate that this is a manipulation that induces a set of addiction-like behaviors. So if this whole choice phenotype that we've identified really has something to do with addiction, the prediction would be that if you induce dependence passively using this model, then that should increase the proportion of the population that chooses alcohol over natural reward, right? And this is unpublished. We're now venturing into unpublished territory because this is such an important piece of work. We really want to elaborate on it, but that's exactly what happens. So you do this for 7 weeks. Let me point out, this is not acute withdrawal. This is we let withdrawal dissipate for over a week. We're looking at the persistent changes, and you can see that that this 15% that you had before exposure is bumped up to about 50%. One reason we're so excited about this research is that in addition to allowing us to uncover the underlying mechanisms of what happens as individuals, might allow us to uncover what happens in individuals as they develop addiction. It also has the potential to teach us something else. Please note, 50% of the individuals seem to be protected. They get the same exposure and they don't shift. And I would very much like to know what it is that confers that resilience, and that is a major project that we're embarking on right now. Okay, so sanity checks out. This is tremendous amount of work. It's probably, if you went off to pharma and proposed this as a model for target ID and validation, they would laugh you out of the room. It probably has exactly all the properties you don't want in a model for those purposes, but what the heck. This is the one time in my career I said, doesn't matter how much work it is, doesn't matter how many rats it takes, because this is important. So we decide to use this for our target ID efforts, because after all, in the end, what I'm interested in is drugs. And to have drugs, I need targets. So for target ID, you could do any number of things. Differential gene expression analysis has been on the menu of our lab since 25 years. If I tell you that the first differential gene expression analysis that we did was by the postdoc and me dragging a 1.7 meter tall differential display gel into the lab, running out the randomly primed things on the gel and then cutting out the bands that we recognize as being differentially displayed with a scalpel, then you'll know how old I am. Since then, we've done, we've done Affy, we've done Illumina, we're now mostly doing RNA-seq. I can tell you it's still the case that nanostring is my favorite. And the reason it's my favorite is, yes, it's a medium throughput platform. We get 400 probe sets onto a chip, but what we get back is so quantitative that I fell off the chair when I saw it the first time, and I'll show you some of those numbers. So that's my favorite, and I'll just stop going on and on about that. So what you see here is a differential gene expression screen through 6 usual suspect regions of the brain. And remember, we're looking now at the pre-existing patterns, because these guys segregated with regard to the behavior spontaneously. We didn't do anything to them, and we control for exposure. One of the nice things of the choice model is that in the end, they really don't consume that significantly different amounts of alcohol. They just have a chance to reveal their choice preference. So this is not contaminated by effects of alcohol. This has to be pre-existing vulnerability. So 5 out of the 6 brain regions, there's nothing. I mean, if this had been Affy or RNA-seq, there'd be hundreds of transcripts going up, hundreds going down, and of course it would take me 2 years to figure out what of this that's noise and what, if anything, that's not. Very different with this. 5 out of 6 regions, there's essentially nothing, and then the amygdala pops out. And then as we do the pathway analysis on the amygdala, this is really cheating. This is an afterthought, because the actual pathway analysis was carried out by Eric sending me the Excel spreadsheet, me sorting on p-values and fold changes, holding it up and saying, there's a GABAergic gene, and number 2 is a GABAergic, and then we did ingenuity pathway afterwards. And as we did, let me just blow up this module for you. So we have this module of downregulated GABAergic transcripts, and at the root of it, we thought immediately must be the GABA transporter GAT3, because it's easy to imagine if you downregulate the expression of the transporter, then you impair GABA clearance, then you'd expect elevated levels of extracellular GABA, and secondary to that, you downregulate a bunch of transcripts for subunits of the GABA A receptor, etc. Right? So this is the one. So reviewers asked us to validate our nanostring results using qPCR, and I guess my way of stating this is, gee, I never knew that our qPCR was this precise, because in my mind, basically what the nanostring data do is they validate the qPCR, but you can view it either way you want. 0.9 correlation between those two. So that's pretty cool. There's a couple other molecules I don't have time to talk about that are kind of interesting, but GAT3 is, as you can see, dramatically down. So the prediction I told you is excessive GABAergic inhibition due to impaired GABA clearance. That's exactly what you see here. This is slice electrophysiology. I guess the most revealing slide is here. Once you drop some bicuculin onto the slice, you uncover this, this excessive inhibition that was there previously in the alcohol-choosing animals. That was a very interesting convergence, because across the street from here, my good friend Marisa Roberto and I guess her mentor George Siggins had for the longest time trying to tell us that there's something wrong with GABAergic transmission in the central nucleus of alcohol-dependent rats. And I trust Marisa with my life, but I couldn't figure out what it was, and here we stumbled upon what turns out to be a complete convergence with what Marisa's lab has been doing. And it's really nice to see in this age when people question whether anything is real and can be replicated. This is just a completely different way of coming at the mechanisms that Marisa had been studying in her work. Okay, so that's all nice, but it's all correlational, right? And the days of correlations of publishing good papers with correlations is over. Going to nail down causality. So, and this is the part about science that I love and that I hate, because of course the really important experiments, you got to generate a strong testable prediction, and most of the time when you put it to test, Mother Nature says, nah. And that's the way it's going to be. So the prediction here is If this is causal, then taking the rats out of the majority that doesn't choose alcohol over natural reward and knocking down GAT3 in their central nucleus of the amygdala should convert them fully or partially into rats that actually do choose alcohol. So Estelle Barbier in the lab developed a very unusually efficient AAV shRNA vector. You can see just how efficiently it knocks down the transporter at the level of RNA, and then of course the protein turns over much more slowly than the transcript, so we would have to go much further out to demonstrate equal magnitude of knockdown, but already after a relatively short time you're down by 50%. This is, by the way, mass spec, and just a funny aside, we went over 7 different GAT3 antibodies in the course of running the study. 2 or 3 of them had been published in really good journals. We did get bands on our westerns. The only problem was we actually bothered to cut out those bands, throw them into the mass spec, and there was no GAT3 in them. Okay? So, which made it even more fun when one reviewer at some point asked, this is all nice, but why don't you confirm your protein results using traditional Western blot? Anyway, this is the scary experiment, okay? So yeah, the knockdown vector works. Let's do the in vivo surgery, and of course this is really stressful. They recover, they resume baseline responding, and there's no difference whatsoever, and that's when you have to pretend to be a good mentor and not show the postdoc that you're just as nervous as he is, and remind him, hey, wait a second, these vectors take about 2 to 3 weeks to fully express, and by the time it's expressed, so 2 weeks is here, right? So prediction supported. Okay, this is causal, this is great. Once again, very friendly reviewers saying this is wonderful, and then suggesting, I think, 6 or 7 additional rat models in which they wanted us to test whether GAT3 really was causing elevated alcohol consumption, including P rats, SP rats, and a few other things. We looked at this and said, that's not what matters, right? What matters are humans. We got postmortem brain tissue from the wonderful Australian brain bank that the NIAAA has been supporting forever. Those people are just amazing. In the course of doing this, we figured out that Dane Mayfield was doing that in Texas, was doing something very similar, so we decided to team up. Ultimately, we did the RNA sequencing together, and you can see that in the central nucleus, just of humans with alcohol use disorder, you see this downregulation, and it's as selective as it was in the rats. So at least, had it been just the human data, it's impossible to know whether this is a pre-existing cause of developing the addictive disorder, or it's just a consequence of exposing your brain to lots of booze. This is what shows the strength of combining preclinical and clinical approaches. We think that this could be both a cause and a consequence, of course, because it could be feeding into each other, but based on the rat data, we think that it's more likely to be a pre-existing risk factor than anything else. Okay, so remember I told you how the choice phenotype pulls out with it an aversion-resistant alcohol self-administration phenotype, but we can, of course, come at it from a different angle and just look for the aversion-resistant self-administration per se. Turns out that the guys that are the choosers, the alcohol-choosing rats, they're a subset of the guys that are the aversion-resistant alcohol self-administrators. So if you do it this way, and this is Essie Domi's work that we think this is the next big story. So Essie's been working very hard on this, and she gets around 30% of rats that will continue self-administration despite getting signaled shock. And again, this is not nociception, it's not this, it's something about those rats just pursuing alcohol like that. Just a hint of the technical trick. So what happens when you turn on the shock with alcohol is all rats drop. And then you have to keep going for a few days. And then the ones that are aversion resistant, I could say compulsive, but I don't quite like that word because that has different connotations for psychiatrists. Then they sort of come back up, and then they stay, and then it's a very robust phenotype. Okay, so we have that, and so we Essie and a colleague of her carried out the massive force mapping efforts. So what cells are active in association with this aversion-resistant alcohol seeking? And if you think about it, this is a nightmare of an experiment. Again, we seem to excel in those because now you have to have yoked control groups, because obviously we're not interested in the cell populations that get activated by virtue of the rat receiving electric shock. We want those that are associated with a specific aversion-resistant alcohol seeking. So there are actually 4 groups in this experiment. Once it's done, you could show it region by region. Hey, look, significant difference here. I don't like that because, or at least I have a very hard time making sense of it. So we try to understand what are the networks. And one way of trying to do that is using principal component analysis and see what travels together. It's a very simple way if you compare it to trying to figure out networks in, if let's say, brain imaging studies where you have the temporal dimension to deal with as well, but it's fundamentally the same approach. So 2 nice networks, right? And based on Jeff Schoenbaum's work, based on recent work from Christian Luescher, but also based on the fact that every postdoc wants to have their own little brain region and not be squeezed into the same as their colleague, SE really hoped that the orbitofrontal cortex would be part of the network that's really important for aversion-resistant alcohol seeking. And then there's this other network, and you can see what's in that one, right? So you can see where this is going. It's, of course, exactly the opposite. Even though both networks are turned on, it's only network 1, which is this one with the central nucleus in it, that really distinguishes the rats that are aversion-resistant alcohol seekers from those that are not. The other one doesn't. And this is so this is where the file conversions don't end up pretty, but what you see here is the activity within the central nucleus correlated with the number of responses to obtain alcohol despite signal shock. And you can see that there's the level of correlation is 70% of variance in behavior explained by a cell-based marker. I have rarely seen that, if ever, over the course of my career. So clearly the central nucleus of the amygdala pops up as a major hub in this network here as well. But, I used to think that I know something about the amygdala. That's where I did my graduate thesis, and things were all simple. We all read Larry Swenson, and I think my extremely sophisticated notion was the basolateral lateral complex was this old cortical structure that had descended for a meeting with essentially subcortical, central, and medial nuclei, and those Larry called the motor ganglion of your emotional life. This is a famous review paper, probably 25 years ago, What is the Amygdala, which is, of course, a play of words on an even more famous classic by William James, What is an Emotion? It's gotten a little bit more complicated since, thanks to people like Kay Tai and others. So we know now that there are within the central nucleus these different populations that are labeled by different phenotypic markers. 2 very important populations, as it seems, that I'll come back to in the cartoon in a moment, are labeled either by co-expression of somatostatin here or PKC delta here. So what you see here is the activation that accompanies the aversion-resistant alcohol seeking. So here is the FOS marker for active cells, and then you see the 2 phenotypic markers, and you see the overlay, and every cell that gets activated is a PKC positive cell. And no cell that gets activated is a somatostatin positive cell. And this is just a quantification of this, and you see the foot shock, the importance of the foot shock yoked group. Being shocked does bump up the activity of these cells a little bit, but the really important increase is the one that accompanies the aversion-resistant seeking. Okay. So things used to be simple, not so much anymore. So these are the 2 populations. These are the PKC delta cells in the centrolateral division of the central nucleus, and this is the somatostatin cells. And Lichty, in a series of beautiful papers, mapped out these and characterized their function, but this was all in the context of fear learning, right? So the PKC delta cells, well, let's start with the somatostatin. They're supposedly fear-on cells, and these guys are fear-off cells. So, it's not immediately obvious how that links to aversion-resistant alcohol seeking, but I have a It's a very speculative interpretation that I'd be very curious to discuss with you. What happens, what Lichty sees when he activates these fear-off cells is basically they gate the output from the central nucleus. And in the context of fear learning, what that means is that when previously animals were freezing due to exposure to shock-associated stimulus, now that freezing will be turned off, hence fear off. Well, what if aversion-resistant drug seeking is basically a failure to inhibit an appetitive impulse? So you get all this evaluative network going to, it's something that's worth my effort, you prepare to initiate approach behavior, but then there is another network integrating the information you've acquired that tells you that something bad will happen, and this is prevented by a signal from this circuitry. Now, if you activate the fear-off cells, the previous state, not going forward, would be the equivalent of the freezing, while when you turn on these cells, you eliminate that stop signal, and now those animals in which that happens will actually approach and obtain the reinforcer at the expense of receiving the punishment. So big science, speculation, but hopefully you'll agree with me, potentially interesting one. More practical importance. So this is Rainer Spanagel wrote up a commentary to our choice paper, and nicely illustrated. So supposedly GAT3 is down as in rats choosing alcohol over natural reward. So all this stuff I already told you about. You could try to rescue this pathology by drugs, and the most straightforward strategy would be, well, let's see, can I get a drug to bump up the expression of GAT3? I'm not aware of precedent for that type of drug action, although there's some stuff coming out with chaperone proteins for dopamine transporters, so who knows, but that's beyond the limits of what I can understand. You could come up with perhaps less unrealistic approaches, being having an allosteric modulator turn up the efficiency of the number of transporter molecules that are there. Theoretically feasible. Every medicinal chemist I talk to about it laughs me out of the room. You could instead, says Reiner and we, capitalize on the fact that presynaptically in those synapses you have GABA B receptors that control GABA release. And of course, a crude way of compensating for the excessive GABAergic inhibition that results from impaired clearance is to put more of a brake on the release. So basically, you'd expect GABA B agonism to some extent crudely rescue this pathology. That it does. We have a drug that does that. It's called Baclofen. It's been on the market for the last 5 decades, I believe. She stuck the tiny amount of it into the central nucleus of the amygdala, and you can see that, so here she calls it compulsive. Told you I don't like that, aversion resistant. But it's, but GABA B activation in the central nucleus really selectively suppresses the aversion resistant alcohol seeking. So that's nice. It also selectively inhibits the activity of the PKC delta neurons that normally are activated in conjunction with aversion-resistant alcohol seeking. So that matches up. Still haven't demonstrated that it is this activity and its inhibition that is causal for the aversion-resistant phenotype. So you can imagine what experiment that remains for that big paper. And so we're trying to approach this using the Brucehopes Fossil-Z rats, and basically using down O2 will turn these cells on, then kill them, and that should prevent the aversion-resistant alcohol seeking. So that experiment is underway. So, but come on, Baclofen, it's been around forever. We've heard about this guy who cured his alcoholism, and I'm allergic to doing science by anecdotes. When I saw this, I wasn't particularly thrilled. But then, by the time a certain Lorenzo Leger, whom I then had the pleasure to recruit to the intramural program, actually published a Lancet paper showing very elegantly that in people with severe alcohol dependence, Baclofen really had beneficial effects. These were people who had liver cirrhosis, they couldn't get naltrexone, so they attempted Baclofen. At the same time, J.C. Garbut was running a Baclofen study in the US. It was negative. Both these guys are fantastic scientists. Rather than starting a fight over who's right, who's wrong, they started looking at the differences between their studies, and they figured out very quickly that the severity of dependence was the single most striking difference between their studies. JC had recruited the way most American studies do, by newspaper advertising. The Italians had recruited right out of a stream of very heavily dependent people, basically on the waiting list for a liver transplant. So JC, being the wonderful guy he is, went back and said, I got to run another study with more severely dependent people. Rumor has it he has completed that study and it's positive. So it's interesting because all the stuff we've been seeing in the rats indicates that this mechanism really gets engaged as dependence develops, and that would support the human findings. But Baclofen is great for proof of principle. It's great for some patients. I have a bunch of patients on Baclofen. Some of them really benefit quite a bit, but you can imagine if day after day you keep kicking a G protein-coupled receptor with an orthosteric agonist, we know what to expect. It's going to downregulate, you're going to have tolerance, people are going to escalate doses, and you're going to get in trouble. And that's exactly what happened. So in France, there's been a number of deaths in situations like this, which is the reason why the European Medicines Agency turned down an application for Baclofen to receive an expanded indication for alcohol addiction. So this is good for proof of principle. It sort of supports this whole conceptual framework, but it's really useless for bringing forward a new medication. However, in a different part of my brain and my lab, totally serendipitously, a small Swiss company called ADEX approached us in my final days at the NIH and said, hey, we have this positive allosteric modulator for GABA B receptors. Would you care to evaluate it because we think it might be useful in alcohol addiction? And we said, sure. And we ran it through our standard battery of all those completely flawed animal models that I told you not to trust. And it worked. So, we got some money to support the lab. We published a nice paper, and that was it. And then, by the time we stumbled upon the choice findings and the role of GABAergic dysregulations in the central nucleus, we basically did this. And we ran back to the chemical cabinet, pulled out what little we had left of this substance, pulled some scrap rats that were leftovers from a choice experiment, and said, let's see, this should normalize choice behavior. Well, you can see what happened, right? So, complete reversal of the pathological alcohol choice preference in the 15% minority that displays it. So that's it. That's what I have to tell you. Just to summarize, I think at least the models that my lab has been using seem to have had limitations enough that I have so far not been able to bring forward any medications I could be proud of once I retire. So thinking about these things, we note that most rats will not do some of the things that are perhaps most important in clinical alcohol addiction, such as choosing the drug at the expense of natural rewards and pursuing taking the drug despite knowledge of adverse consequences. But we do have a minority that displays those traits, and it seems like that minority might offer a fruitful foundation for target identification efforts and for discovering new mechanisms. And one of the first to pop out seems to be GABAergic targets that I already talked about. Just zooming out and in a somewhat bigger perspective, I think there's a broader message here, and I don't want to sound too philosophical, but if you think about it, patient comes into my office, I diagnose alcohol addiction, and I pull out the notepad and I offer a script for naltrexone, and I basically, with that, I'm telling the patient, here's something that will take out the fun from using alcohol, which might be the last thing that was fun in your life, and if you had anything else that was still somewhat fun, it might actually attenuate that as well. great incentive for the patient, right? And then we complain that compliance is crappy and that clinical uptake is bad. More broadly, I think this research and combine that with clinical experience points to the fact that looking at the objective of addiction treatments as being only to reduce drug taking is fundamentally flawed. I think unless there are healthy options available to the patients, it won't matter what miracle drugs we try to come up with. We have to be aware of the importance of not only the choices and the choice preferences, but actually the availability of healthy choices, jobs, housing, social integration, and that tends to get forgotten. So with that, I'll stop and I'll take any questions.
Is anybody remote or not?
Yeah.
Are there any questions? I'm going to maybe get us started then. Get over here. With the science paper, Eric's science paper, and you have the preferring and the non-preferring rats, I think those are the terms used for them. And I know we emailed about this, so forgive me if I'm asking you a question that you've already answered by email, but have you bred the animals to see if that characteristic breeds true, that it's a genetic trait of the animals, or not? Because it could very much be an environmental or some other cause, and it's important to know the difference because there's a lot of things you could do if it was in fact genetic. Can you remind me?
So I wish I could say yes. So the choice was either having animal housing enough to do that, or having animal housing to allow Eric to establish an independent lab. And so, because he just landed a startup grant, and so we don't have the housing currently to do that breeding, and it's very high up on the list that I would like to do. But so far we have not. You know, as a surrogate measure, we try to sequence some of the interesting stuff here. Most importantly, of course, the promoter region and then some other regulatory regions of the GAT3 gene itself. But that's a crappy surrogate for what you're asking for.
Right. Yeah. Yeah. Okay. Mark. Yeah.
Just a little historical curiosity. This being the Markou seminar, the little company called Adex that was founded by two guys, one of whom was a postdoc with Athena.
I did not know this.
Yeah.
I had no idea. I can tell you as much, so that's wonderful to hear. And secondly, life was a lot nicer and simpler when they held the molecule. Of course, in part based on these data and others, Indivior licensed in this molecule. And I can tell you that since then, life's been less fun. And we ended up, in the end, having a friendly neighborhood chemist make close to 10 grams of this stuff for us because it was so difficult to deal with the company. But the Adex people were fantastic. So that's nice, really nice to hear.
Okay. Oh yeah, great. Okay.
Thanks for an interesting talk. So you showed the mechanism which is related to the severity of addiction, but are there any trials to look for the different subgroups of in the animal studies of rats, for example, you know, those who had more troubles with some stress reaction or more impulsivity, and see if there are different subnetworks involved in the same alcohol use disorder?
Yeah, so something funny had, so it's very primitive, but you know, part of the standard battery that I referred to involves relapse studies, and basically you see among the main categories of relapse triggering stimuli is priming doses and drug-associated cues, and in my mind they're very closely related, and then you have stress as the third. So we predicted, based on all these things, that this would be specifically a mechanism for animals where stress is the negatively reinforced alcohol seeking is the primary driver of the behavior, and that possibly the GABA-B PAM would attenuate that, but not cue-induced reinstatement. To our surprise, it attenuated both. So I don't know what that means. Other drugs, if you take, for instance, a kappa antagonist, which we had in our lab and published in Neuropsychopharm, just sort of paper prior to this, to the GABA-B PAM, that very nicely differentiates so that it blocks stress-induced reinstatement but does not block cue-induced reinstatement. So yeah, there are attempts, but they're very crude, I would say.
Okay, and the second question is, in this study from 1993, where there is, the percentage of problematic
4,
users,
I guess. But
yeah, in case of alcohol, it was something around 9%, as I remember.
15.
15?
15, which
In
just
humans?
happens to be exactly the same number that we get in our rats. I think
Okay.
that's totally a fluke, but it's a nice fluke for us.
Okay.
So what about it?
No, I remember that it was 9, and I was asking why is it 15 in rats? It's the same thing.
So I got to tell you this. We're cheating. So this is not an absolute number. If you can play around with this parametrically. If you start devaluing the reinforcer, which you can easily do by systematically delaying its delivery, you will shift this number around.
Okay.
Not a whole lot, but it can range anywhere between 10 and 20. It just, with no manipulations, it ended up being 15 on first attempt. That matched up with the human data. We said, oh, that's great. We will claim that it's the same number. But we know that this is not an exact science. The important point is there is a significant minority that has this pre-existing vulnerability for this syndrome of traits.
And the last one, what are your guesses for this 50% who are completely resistant for development?
I have no clue. Do you have any?
No, but maybe you have any hypothesis.
Honestly, no. It's almost like there's very little, if any, prior research to sort of base any hypothesis on. Most research has been focused on figuring out the risk factors and the mechanisms that promote alcohol seeking and taking. So I just don't know.
Okay.
Yep. But that's the beauty of the data-driven approaches. Doing gene expression profiling will allow us to, and that's the situation where we'll absolutely do RNA-seq to cover the whole transcriptome. Yes.
Very nice talk.
Thank you.
I was wondering, based on the lesson that you learned from Eric's data, ACEs data, are they done with a choice of the animal self-administering alcohol and sucrose and then food shock, or?
No, we're cheating. We got to be somewhat practical, So for now, we're sort of pulling these two phenotypes apart and studying them in isolation. We will have to put it together at some point, but I just pray that point will be later rather than sooner, because it's just so much work.
Because the reviewer will ask you, what about in the natural reward or in the sucrose, based on?
Yes, but I can tell you. So okay. So we do consider this enough, at least, to include the following critical control. Is this specific for alcohol, or would they disregard concomitant punishment even when self-administering a saccharin solution? And the answer to that is no, absolutely not. That all those rats get perfectly suppressed when they're self-administering a saccharin solution, but remain compulsive-like, if you will, for alcohol. So that's encouraging, at least.
So the consecutive question was, do you have any data that correlate the number of rewards with the blood alcohol levels?
Oh yeah, they consume everything that they get. That's actually, I think George Krug was the first who bothered to get that kind of data, and it's a very nice correlation, and we don't do it in every experiment.
Because as a person who was doing these experiments before, I have struggled with it by myself, and I've seen that animals jump in the receptacle where they receive the alcohol or food or whatever, trying to avoid the food shock.
Oh, yeah.
So it looks like the animals are self-administering more. In reality, they don't feel the food shock because they jump. So I was just wondering if after the session you guys check the alcohol level.
Yeah, we have a 0.8 correlation. Very, very robust. One trick to this is I didn't go into this, but we take them up to FR3 at some point. So we train them step it up to FR3, and by the time that's done, it's very hard for them to cheat.
Thank you.
Yeah, were you doing FR1?
I was doing upper 5.
Oh,
That's better than 3.
yeah, that's better than 3. Okay, then I don't know.
Good cheaters here.
Okay. All right, have I totally exhausted everybody? No, please.
Yeah, great.
Great talk. You mentioned that baclofen's acting on the presynaptic autoreceptors. Is it possible that it's actually also acting on the postsynaptic GABA B receptors?
It's possible. So I was just... Okay, here's an indication why it is likely that it's the presynaptic receptors that are important, and this is Marisa Roberto's work, and it's kind of takes us into stuff I skipped over. So, you know, as you induce alcohol dependence, you get excessive GABAergic inhibition in the central nucleus as well, right? So that was the convergence I did talk about. So you might think that that's the result of the same thing that you see in the pre-existing vulnerability, namely downregulation of GAT3 expression. It is probably not. You can, you know, it doesn't functionally. A number of dysregulations can converge on the same functional output. So it looks from Marisa's work like what happens in the rats where dependence is induced is a downregulation of the presynaptic GABA B receptors, and in those guys, it is that which prevents released GABA to auto-inhibit and therefore gets out of control. While in our data, GAT3 is not downregulated in the alcohol vapor exposed rats. So that's an indication that it is the presynaptic GABA B receptor that's critical here. Yep. Exhausted now?
Can I add 2 more?
Of course.
First of all, you may have said it, but what is the distinction between the ADX drug and Baclofen, or do we not know?
Oh, we do.
Oh, yeah.
So ADX is a positive allosteric modulator.
I see, okay.
No tolerance, no sedation.
Clear. Yeah.
Maybe I didn't mention, sorry.
Okay, yeah, no,
Yeah.
if you mentioned, I missed, yep. And do we know how GAD3 is getting changed or why it's different, or is that a big, big mystery? Do you have an idea of why that is? It could be...
No, because for...
You hope it's the inherited regulatory differences? I hope it's inherited regulatory differences, but we don't know?
We don't know. We're doing the sequencing right now for practical reasons. It's gotten delayed several... Well, we don't even know that there are functional variants in the promoter region
right.
So we're trying to get those sequences as we speak. So far, no clue. And if that doesn't pan out, we're going to do a methylation scan. And if that doesn't work out, I have no idea, and we'll ask you for advice.
If you bred them,
yeah.
maybe just see if the offspring inherited that difference. Yeah.
Do you have some animal space we could move into?
Garage, right? Don't you have garages in Sweden?
Yeah, no, you know, if you can provide us with advice on how that short-term breeding should be done, that is certainly...
We should restart that conversation.
Yeah, we should absolutely do that. Yes.
Good.
Yes. Okay, good. Done?